Welcome back. We are starting with chapter 35, and we're going to discuss about infection and pathogenicity in this chapter. So we are going to talk about the process of infection first. So the infection, we are just trying to develop some of the knowledge about some of the definitions. The first thing that we are going to define is what is a host. So the host is a larger organism that supports the survival and growth of a smaller organism. And here, by all intent and purposes, the host is basically the human beings or us. So the infection is, on the other hand, is a microbe growing and multiplying on or within a host and may or may not result in overt infectious disease. So many a times, there may not be any outcome of the infectious disease, but the person could be still infectious to other people, and they are known as the carriers. On the other hand, the infectious disease is basically any change from a state of health, and part or all of the host is incapable of carrying on normal functions due to the presence of a pathogens or its product. So these are some of the very basic definition that we have already seen. Then we are going to define a pathogen, and a pathogen is an organism that causes a disease. Now, this pathogen for the human beings, it could be a virus, it could be a bacteria, it could be a fungus, it could be a protist or protozoa. These all can be pathogens. And even there are some large eukaryotic pathogens, say for an example, the worms or the nematodes, and those could be called as a pathogen as well. So on the other hand, the opportunistic pathogens may be a normal flora that causes a disease when it gains access to the other tissue sites or when the host is immunocompromised. A classic example of these opportunistic pathogens, say for an example, that we have a lot of these E. coli in our gastrointestinal tract, particularly in the colon area. They do help in digestion of our food material. They have a safe harbor, safe haven. So they also produce some of the vitamins that are essential for us. So we do have a mutualistic relationship with this E. coli in our gastrointestinal tract, particularly in the colon. But from the gastrointestinal tract or the colon or the large intestine, if the E. coli move into the urinary tract, then this is basically when it gains access to other tissues. That means it moved to the urinary tract and it can cause infection in there. So these are called the opportunistic pathogen. On the other hand, there is another group which is not explicitly written here. We call them as a primary pathogen. So the primary pathogen, in contrast to the opportunistic pathogens, are that no matter what, they will always cause a disease. And that is very common in many of these. Say, for an example, Clostridium botulinum or say Entamoeba histolytica. These organisms or the pathogens get inside our body. They will always cause a disease. So those are the pathogens. Primary pathogens always cause a disease. Opportunistic pathogens, only when they are displaced from their native place, they can cause a disease. A pathogenicity is the ability of the pathogen to cause a disease. And there is also another definition, is a virulence, which is the degree of harm or pathogenicity inflicted on the host. So let's take a look at that. Say, for an example, the rhinovirus, which causes common cold, is a pathogen, whereas the influenza virus, which also causes flu, is a pathogen. So both of them are considered as pathogen. So what varies in between these pathogens is the virulence. Say, for example, we all know the rhinovirus, which causes common cold, the degree of virulence is much less. You can do all your common functions. You can go to school, you can go to your work, and there is no problem with that. But on the other hand, for the flu, which is caused by an influenza virus, the virulence is much more. And at that time, you are supposed to stay indoor and not go into the public when you have a flu, because the virulence of that virus is much more severe. And now you know, of course, that this coronavirus outbreak is going on in China, which is spread all over the world to some extent, and very, very virulent. So the degree of harm inflicted on the host, what it means by the virulence. So extracellular versus intracellular pathogen. So when we are talking about the extracellular pathogen, they contact a host and survive within it, and it causes a disease. And extracellular pathogens will grow outside the host cell, say in the tissue and the fluids, And a classic example for that is the Yersinia pestis. This actually is a plague-causing bacteria, which also caused the Black Death in the Europe in the Middle Age, which probably wiped up almost half of the population of the Europe as such. Extremely virulent organism, and these grows outside in the tissue fluid and they never get inside the cell. On the other hand, there are some intracellular pathogen, and in the intracellular pathogen, there are two types. They multiply within the host cells, and what we mean by these, that these facultative intracellular pathogens reside within the cells of the host or in the environment, but also they can grow in the pure culture without the host cell support. Say, for an example, if we just take an example of a bacteria, like say Brucella. So the Brucella abortus, they can infect the macrophage, they can infect the neutrophil and also the trophoblast. This is the cells of the tissues around the embryo or the fetus, the inner lining of the uterus, and they infect all these areas. But Brucella, you can easily grow in the laboratory as well. Another example of that is the histoplasma. So the histoplasma is another example where it can grow inside the cell of the host, but it can also be grown in the laboratory. On the other hand, there are some specific obligate intracellular pathogen, and these can only grow inside the host cell. A classic example of that is, say, for example, for the chlamydia. On the other hand, there is a large group of organisms which are known as the rickettsia. These are bacteria, but they are specifically in the group of rickettsia, and these rickettsia can only grow inside a living cell. So they always grow inside a cell and they cannot grow outside a cell. So those are obligate intracellular pathogen. On the other hand, we always know the classic example that every virus is always obligate intracellular pathogen. They do not have a living entity outside a living cell. On the other hand, say for example, Plasmodium, which causes malaria, that also need a living cell for its development and differentiation. They can live outside the host, say for an example, of the mosquito when they are being transmitted, but they will need these red blood cells or they need to go into the liver cells for their growth and differentiation. So now let's take a look at the course of an infectious disease, and this is a typical curve which shows the infectious disease. So for example, that first a host, which is right here, we think about the human being, we had initial exposure to the microbe. So after that, we have an incubation period, and this is the period which is after the pathogen entry. And this is completely silent, this area. That means you do not feel anything during the incubation period. Now, this incubation period could be as small as, say, 12 hours, 6 to 12 hours, say for an enteric bacteria, or say Clostridium botulinum, it could be 12 hours. On the other hand, there are some viruses which it could be 2 days. Some viruses, it could be 7 days. There are some examples, there could be the incubation period could be even 3 weeks. So that's the incubation period. The next area is known as the prodromal stage. And here you have an onset of signs and symptoms, but this is not clear enough for diagnosis. So just think about that. Just before you get a common cold, you probably a day before, you do get some kind of a itchy nose, itchy eyes, and you are just getting something, but still you don't get a common cold. When you say to the other people that I'm coming down with something, that is exactly what a prodromal stage means, which is also known as a vague symptoms, some vague signs and symptoms. After that, it is followed by an illness period, and usually this is the disease is the most severe and the sign of symptoms. And of course, you can get into a condition which is diagnostically possible to determine that what is the cause of the disease. And after some time, this disease may level off, means either by the intervention of your immune system or by the therapeutic agents, this illness should be checked after a certain period of time. If it cannot be, it can lead to death. And then it is followed by a convulsence, which is a sign and symptoms seems to disappear. At that time, although the person can shed that pathogen, the person could be infectious, and contagious at this period of time as well. So you can easily think about that simple example for common cold, that you have contacted the virus, you have the incubation period, then you have this prodromal period, just a day or two when you think that you are coming down with the something, then you have approximately about 7 days a week of the common cold, then is the convalescence period when you recover from that. Many of these diseases which cause mild diseases, our immune system is good enough to take care of these. On the other hand, there are some where just our immune system is not capable of taking care of the disease within a certain period of time, and we need therapeutic intervention. So here are some definition. A signs is an objective change in the body that can be directly observed. For example, say fever, rash, these are the signs. Symptoms, on the other hand, is a subjective change experienced by the patient, and the symptoms cannot be determined by the other people until and unless the patient actually tells other people. Say, for example, the fever can be felt by another person or by using a thermometer. A rash can be visually possible to see. On the other hand, say for an example, a pain or a loss of appetite or a headache, these are the symptoms, but it cannot be determined by the other people until it is said by the patient or it is conveyed by the patient or communicated by the patient. On the other hand, a disease or the syndrome is a set of characteristic signs and symptoms for a particular disease. And there are several different types of disease or the syndrome. You can see that you can have a gastrointestinal diarrhea. On the other hand, you can have a characteristic, say for an example, when you have a strep throat, you do have these characteristic white tonsils marks in there, and it is almost clearly the doctor can see and diagnose that probably you have a strep throat. And for an example, chickenpox, when you have these rashes all over your body, which is a very characteristic, you can diagnose that this is a chickenpox. There's the same way that when you have measles, there is a spot inside your throat, which is known as a Kopik's spot. Those are the characteristic sign and symptom for some of the diseases. The next part, which is importance of resources, the host provide the pathogen for the protection, nutrients, and energy to use. And the infectious agent develop a mechanism to access and exploit the host. To survive, this pathogen must also devise methods to move onto a new location or host when necessary. So on the other hand, if you remember that from the last chapter, we talked about this, that no matter what, the symbiotic relationship with a pathogen to a host is always parasitic. That means the pathogen is always going to do harm to the host. So that is basically the relationship. So we are looking at after that what are the events in the infection and the disease. So the transmission from a previous host or a reservoir to a new host is the first start or the event in the disease. So the virulence of an organism, number of invading organism, and the presence of adhesion and invasion factors will affect the success of the pathogen to establish and start an infection. So the organism outcompetes the resident microbiota for resources and survive host defense mechanism. So this could be, if it is a gastrointestinal organism, that can essentially outcompete the resident microbiota and compete for survive the host defense mechanism. Say for an example, if we just particularly think about this, that you have already heard that there is an E. coli strain which is known as E. coli O157, and this particular strain, which is this O157, that causes the bloody diarrhea. And sometimes these lettuce or spinach is recalled from the grocery store due to the presence of these E. coli. These are some of the rogue E. coli which actually outcompete our own E. coli that is present in our colon or the gastrointestinal tract. So this is some example of those. Now, on the other hand, the disease occurs when the organism produces molecules that directly damage the host cell or stimulates the host immune cells to destroy infected tissues or alters the host cell genome to affect the normal function. And we're going to see a lots of examples of this. The next part that we are looking at, that the transmission and entry to the host and how the pathogens are successful in doing so. And the sources of pathogen can be animate. like means that they can be other humans or the animals. And the infectious is passed from an animals to the humans are known as zoonosis. This is actually a very important term. And we know that there are many examples of this. Say, for an example, with just a couple of examples, for the West Nile viruses, the water birds are actually the reservoir. So from the water birds, the mosquitoes carry those and spread it to the human beings. And the human beings are just incidental host. Really, the host is not human being. They are the water birds. And horses and the human beings are just the incidental host. On the other hand, you can see that it could be inanimate as well. Say, for an example, say for Vibrio cholera. Vibrio cholera, the water body can be contaminated by the Vibrio cholera, and when you drink that water, you can get the infection from there. On the other hand, the zoonosis, classic example for zoonosis is the rabies. That is the most common. You can see that right here. The rabies can be carried by the cats, dogs, raccoons, skunks. These are potentially rabid animals and can carry the rabies virus on the other hand. On the other hand, say for an example, the reservoir could be a natural environmental location in which the pathogen normally reside. So this could be a water body which could be infected by some particular pathogen. On the other hand, the reservoir could be the human. It could be animate or it could be inanimate. Both could be the reservoir. So remember that an animal or the humans could be the reservoir. Also the water, food, or any source could be a reservoir as well. On the other hand, the vector or the biological vectors are the organism that spread a disease from one host to the other. So there are two types of vectors. One is known as a mechanical vector. So a mechanical vector is that, think about that you have this housefly that is sitting on some reservoir which contains microorganisms which are pathogen. And then it sits on a food that you're going to eat, and they are mechanically just brushing off from their body some of the pathogens that you can eat and get ill. So these vectors are called the mechanical vectors. On the other hand, there are some other vectors which are more important, are known as the biological vectors. These biological vectors are very specific for a particular disease. So if we just take a look at one at a time, say for mosquitoes, there are some particular mosquitoes like Anopheles mosquitoes which carries the malarial parasite. On the other hand, see this Aedes aegypti mosquito, which is responsible for Zika, chikungunya, West Nile virus. All these viruses are carried by these Aedes aegypti mosquito. Then we have ticks. Let's take a look at a very specific one. Say, for an example, there is a particular tick which is known as a deer tick. And this deer tick actually carries a particular bacteria which is known as the Borrelia burgdorferi, which causes the Lyme disease. On the other hand, fleas are very important. There are some specific fleas which carries the Yersinia pestis, which causes the plague. On the other hand, there are some mites which also can carry that. Or there are some biting flies. Say, for an example, the biting flies, classic example for these is the tsetse fly. The spelling is Tsetse, but it's pronounced as a tsetse flies. These are in African continent, and these carries protist, which is a trypanosoma. This is a flagellate, and this trypanosoma can go inside your body and cause lethal sickness, which starts with a sleeping sickness, and ultimately, if it is not properly treated, the person can go into the coma and ultimately can die due to this. So you can easily see that these are all biological vectors. And why we call these a biological vectors rather than mechanical vectors, because each of these carry a particular pathogen and they are fixed. Say, for an example, the ticks will not carry West Nile viruses, or the fleas will not carry the malarial parasites. On the other hand, the mosquitoes will not carry the Borrelia burgdorferi, which is carried by the deer ticks. So they are very specific in association organism they carry and dissipate among the hosts. So the next we are looking at the pathogen transmission, very straightforward, occurs directly or indirectly, and there are four main routes. We're going to see the airborne, contact, vehicle, and vector-borne. And also the pregnant women can also pass a pathogen to the unborn child, and this is known as a vertical transmission or mother to the fetal transmission. So here we are looking at some of these direct contact transmission of the infectious disease. Horizontal contact, like a kissing, sex, airborne droplets when you are close to someone, can dissipate some of these microorganisms. Vector, you can easily see that we talked about that, and the vertical contact is from a mother to the unborn child. On the other hand, you can see that the indirect contact, a classic example for this, we use a word which is called a fomite. So what is a fomite? A fomite is an inanimate object which is a reservoir or contaminated with something. For an example, is a doorknob or say a public faucet. So if a person is infected with, say, influenza or cold or something, say, for an example, the person touched nose or any of the areas or the mucous membrane, and then without washing the hand, if that person touches, then automatically some of these viruses or the pathogens are transferred onto the fomite. So when the next person touches that pathogen gets into that person, and if accidentally that person touches any of the mucous membrane areas, that pathogen is going to get transmitted to the next person. On the other hand, food, water, biological products, these are example of transmission. Also, another important is known as the fecal oral contamination can also lead to these transmission. What we mean by the fecal oral contamination? There are two different ways that once what happens that if there is not a proper washing of the hands, and that can lead to the fecal oral contamination. On the other hand, there is also another fecal oral contamination which happen when there is a catastrophe hits. Say, for an example, there is a hurricane, tornado, or flood. At that time, if the sewer actually spills over into some potable or drinking water bodies, these fecal material which have these pathogens can spill over into the water bodies, and from those areas we can get the contaminated water, and this can be a fecal oral contamination. Main difference between the two, airborne droplet and the droplet nuclei: airborne droplets are much bigger in size, whereas the airborne nuclei are much smaller in size. Dust also can lead to transmission of many of these pathogens. So let's take a look at that. The pathogen's virulence may be strongly influenced by its modes of transmission and the ability to live outside the host. So here we can looking at the transmission alone is not enough for the infection to occur. So here is a definition. A tropism is the pathogen must make contact with the appropriate host tissue, and this is determined by specific cell surface receptor. So there are a couple of examples that some of these pathogens can live outside the human body for a long time. A classic example for this is a Mycobacterium tuberculosis, which causes TB, or say Corynebacterium diphtheria, which causes diphtheria, or say measles virus. All these pathogens can be viable outside the human body for a long time, and they are very successful in transmitting from one person to the other due to their ability to live outside its host. So here we are looking at some of the airborne transmission that we have seen. A droplet is a direct transmission, and they can be up to 2 millimeters in diameter. These are produced when liquids are placed under a force, say for example sneezing or coughing, and they can travel less than 1 meter. So you can see that they cannot travel much. On the other hand, the droplet nuclei causes this indirect transmission, and they are very small. They are 1 to 5 micron diameter, invisible, and result from the evaporation of larger droplets, but may remain airborne for hours or days and travel long distances. Say for an example, if all these people are breathing in a room or in a bus or in an airplane or in a ship or in an enclosed space, all these people, as they are breathing, they are creating the droplet nuclei, and this can travel long distances. Really, the chances of getting the infection from the droplets, which is due to the coughing or sneezing, are much less, where the droplet nuclei, if the pathogen rides on the droplet nuclei, that is much more scary compared to the droplet or the direct transmissions. Dust particle also can cause direct transmission, where the microorganism adhere to the dust particles, also can survive long periods outside the host, can lead to hospital-acquired infections as well. So which are known as a nosocomial infection. A nosocomial infection which is led to hospital-acquired infection. So interestingly, many of the viruses are transmitted by these droplets and the droplet nuclei. Say, for example, the influenza virus can travel through these droplet nuclei. So they can remain airborne for hours or days and travel long distances. On the other hand, say, for example, although extremely virulent, the Ebola virus cannot be transmitted through the droplet nuclei. They are transmitted only by the droplet or the direct transmission, which means the droplet must touch the mucous membrane of another person to get the virus transmitted. So it varies among the different pathogens. The next, what we are talking about is the contact transmission. And this is coming together or touching the source, reservoir, and host. So here, the direct contact could be person to person, and this is a physical interaction of the source and the reservoir. We talked about that kissing, touching, sexual contact, even shaking hands. These are all direct contacts. On the other hand, the indirect contacts, as we talked about, involves the inanimate object or the fomite. For an example, touching a doorknob, eating utensils, bedding, or touching a faucet, or anything like that. These are all your indirect contacts that we have talked about. Say, for an example, if we are thinking about these direct contacts and what those are. Say, for example, HIV can be transmitted through the body fluid. Herpes virus can be transmitted this way, and the staphylococcus also can be transmitted by these direct contacts. On the other hand, there are some vertical ways that from mother to the child, say for an example, the syphilis, which is caused by the Treponema pallidum, that can be transmitted from mother to the child as well, or the mother to the fetus as well. On the other hand, direct contact, even with the animal product or the animals, can also lead to these diseases as well. So for an example, salmonella can be transferred from an animal product or the animal to the human beings. On the other hand, Campylobacter is another example which can also be transmitted by these direct contacts. So on the other hand, the vehicle means that the inanimate material that transmit pathogen. For example, food, water, biological material, fluids, and the tissues, air. So we just talked about that these are the vehicles on which the pathogens basically ride to get to the person. So these are known as a vehicle transmission. And the single vehicle spreads pathogen to multiple hosts. You can easily see that it could be from cosmetics, from the food, from contaminated drugs, from the water. There are so many different vehicles that can lead to the infection. So after that, we are looking at the vector, which is a direct living transmission of the pathogen. And most of them are the arthropods. We already talked about this: insect, ticks, mites, fleas, or vertebrates, example, dogs, cats, skunks, bats. We already talked about these as well. So the pathogens are transmitted by arthropods are often highly virulent. And we have already talked about that. These causes malaria, typhus, sleeping sickness we talked about, which is transmitted by the tsetse flies. There are many other viral diseases which are carried out by the mosquitoes, and they are very virulent as well. We talked about the fleas, which Yersinia pestis, which causes the plague, that is also carried by the fleas. And the important that the pathogen do not harm their vectors. And this is the important part, that the pathogens do not cause malaria to the vector. On the other hand, says the sleeping sickness, the trypanosoma, doesn't cause any harm to the fly, which is the tsetse flies. So that is the important. The vectors are only responsible for carrying the pathogens to the host, but the pathogens do not have any impact or do not cause any harm to their vectors. Here we are looking at some of the examples of the vertical transmission. Occurs to the unborn child, acquire a pathogen from an infected mother. But this is not as common as a horizontal transmission. Babies born with infectious disease are said to have a congenital infection. And this can cause a gonorrhea, which is caused by Neisseria gonorrhoeae. Syphilis causes by the Treponema pallidum, which is a spirochete. Herpes virus infections, German measles. Toxoplasmosis, which is caused by the Toxoplasma gondii. And this is one of the reasons that these toxoplasma comes from the litter box of the cats. And that's the reason that if you have a cat, the pregnant women have to be a little bit extra careful from getting this Toxoplasma infection. So here we are going into some of the very specifics of that, what is known as an infectious dose, 50%, or which is popularly known as an ID50. So the definition is the number of pathogens that will infect 50% of the inoculated host is known as the ID50. This varies largely with the pathogen. So let's take a look at that. So if we are looking at a strain A and the strain B, which shows that the ID50 is 3000, whereas the ID50 for the strain B is 5000. So what it means that if you have a strain A bacteria, 3000 of the cells of these bacteria will cause disease to 50% to the population. Whereas for the strain B, you need 5000 cells to cause the disease to a population. So compared to that, you can compare the infectious dose for each of those, which means the infectious dose is lower for this pathogen compared to the other. So if they are the same kind of pathogen, to some extent, you can compare them in between the two species, that this is actually more virulent compared to this, which is basically the infectious dose is less compared to the other. Now, this ID50 gives you an overview of the process or the infectious dose, but this cannot be really concluded in the sense that you cannot compare bacterial infection with other viral infection or a fungal infection or a protozoal infection, and you cannot compare these, and that will be kind of comparing apples to oranges, because each one of them has a different ways to cause a disease and the outcome. So really you cannot compare different organisms until and unless they are very close to each other. Say for an example, if you have a flu virus, which is an H1 and N1, and if you have another flu virus or influenza virus, which is have H5 and N1, the mode of working of both of these viruses are similar. In this case, you can consider the ID50 value because their mode of causing a disease is very close. But it is very difficult to compare a Staphylococcus infections or its ID50 dose compared to an influenza virus. So that's just an overview of that. But basically, why this is important, that say hand washing or sanitization reduces the number of pathogen, and that is important. Say for an example, in the food or in the beverage industry or the restaurant industry, we have a particular bacteria which is X and its ID50 has 3000. So by hand washing and sanitization, that number should be significantly less in the cleaned utensils. If that number in the utensils after sanitation is close to 3000 and is over, then the sanitization process is not proper. So you have to devise a sanitization process where you can reduce the number of this infectious dose significantly below the dose, so that there could be some bacteria. It is not that it is completely sterilized and devoid of all the bacteria, but it could be a way that it will cause much less amount of disease. So you can easily see right here that as we are getting a lower number at this point, if you look at the number here, so if you just get a particular bacteria X, so about 1000, this is to the power 10 to the power 3. So if we get a bacteria instead of 3000 to 1000, you can see the chances of infection goes down to about, say, 10% or even less. So that is the goal of the hand washing or sanitization process. So the next thing that what we are looking at is the lethal dose 50 or LD50, and this is a dose that kills 50% of the experimental animal within a specified period. And the lethal dose 50, usually we get these numbers, particularly a particular bacteria which are very virulent, or for a virus which are very virulent. Say for example, the Ebola, Marburg, Lassa fever, which are extremely virulent in that sense. So on the other hand, the definition cytopathology is a cellular change, and this can be used to observe the cells in the tissue culture for the death rates rather than the entire organism, where sometimes it is much more difficult to identify that. So we are looking at the virulence factor and their release that can be done by the process of cytopathology in the tissue culture experiment as well. So next step after the transmission, we are trying to get into the process that how these different microorganisms and the pathogens get into the host and adhere and colonize. So the first step in this are the entry and the attachment. And the first important part is what we call as a portal of entry. What we mean by the portal of entry is this is the part through which a pathogen enters the human body or the host. It could be the skin. Most of the time, the skin, unbroken skin, is pretty good resistance. But if there is a cut in the skin or there is an abrasion on the skin, that could be a portal of entry. Respiratory, we already know that as we are breathing, there could be respiratory portals of entry. Gastrointestinal, as we are eating and drinking, we can get the pathogens through the gastrointestinal tract. Urinary genital, this could be the pathogens which can come just by from the touching or from the sexual contact as well. On the other hand, just by touching the conjunctiva of the eye or just by droplet nuclei, some of the pathogen can be transmitted to the portal of entry of the eye. Once the portal of entry is breached or the pathogen gets inside the human being or the host, the next thing that they have to do is an adherence. And the adherence, the first thing is completely, which we call as the mechanical contact. And these are usually very reversible. In this adherence, the first step, the mechanical non-specific contacts, these are due to the hydrophobic interaction, electrostatic interaction, and just by vibratory forces, the pathogens can just get into a mechanical way onto one of those portals of entry. The second part is very specific, where a particular pathogen may have a cognate receptors to which they bind and they get inside the host. So the second part is very specific. And the next part is also that the pathogens also use something which is called the adhesins. And these adhesins is a generic term. They are specialized molecule by which they can attach onto the host. And we're going to talk about that as well. On the other hand, the colonization is a site which is a microbial replication happens on or within the host and does not necessarily result in the tissue invasion or the damage as well. So this is basically the first area to where the pathogen go and try to build their own population. So here we are looking at some of these adherent structures, which we talked about in the last slide. These bind to the complementary receptor sites on the host cells, classified as virulence factors. This could be pili, this could be fimbriae, this could be the membrane and the capsular material. So when we talked about the bacteria, remember that we talked about two types of capsular material. A capsule is very specific and tight, whereas a slime layer is also made up of the glycocalyx, which is much loose. Both slime layer and the capsule could be the adherence factor. And there are also some specialized adherence factor present on the bacterial surfaces. So you can see that right here, the fimbriae are used onto the host cells by the bacteria. Capsules can attach onto the host cells. The viruses use specific receptors, which basically internalize the viruses into the cell. So these are specific cognate receptor, and each of the viruses have specific receptors to get into the cell. So these are some of the adherence mechanism by the microbes. Say for an example, just give one example. Say these E. coli do have these fimbriae. And if E. coli from the colon gets into the urinary tract, we have already talked about that, this gives urinary tract infection. So in the urinary tract, the cells or the bladder, these E. coli with their fimbriae will attach them onto the cells, which are the epithelial cells of the urinary bladder or the urinary tract. And also, they will release some of the toxins which will cause the inflammation of the wall of the bladder or the urinary tract. And that is what we are going to go into the next section though. So here we are looking at some of the attachment mechanism of different bacteria and or some of the viruses as well. You do not have to memorize all of that. Just having a couple of different ones. Say Neisseria gonorrhoeae, which causes a gonorrhea. This is a type 4 fimbriae. Here you have a pili. Treponema pallidum, which causes the syphilis. We talked about that. They have an outer membrane protein and fibronectin. This is a spirochete. Mycoplasma membrane protein. Then you can see that right here, we're looking at some of the influenza viruses. The influenza virus use a hemagglutinin spike protein, whereas the GP120 protein is used by the HIV. capsid protein VP1. So these are some very specific proteins on the surface of the viruses which attaches to a particular receptor. Say for example, GP120 bind the CD4 on the T cells. Hemagglutinin binds to the protein residues on the upper respiratory tract. Specifically, we know that those have a sialic acid in there. On the other hand, the capsid protein BP1 bind to the CD155 protein on intestinal and nerve cells. So each of the adhesion mechanism have a cognate receptor compared to these. So you can easily see that these are the different adhesion mechanism and they can be varied in between the different bacteria and the different viruses. So here, the next part we are getting is the invasion and the disseminates the pathogens into the host. So the infectivity is the ability to create a discrete point of infection. So after colonization, the next part will be the infectivity. And the invasiveness is the ability to spread to the adjacent tissue from the point of infections. And this penetration could be active or passive. So let's take a look at how this can happen. The active penetration can occur due to the lytic substances and alter host tissues by attacking extracellular matrix basement membrane of integuments, intestinal lining, degrading carbohydrate protein complex, disrupting host cell surface. On the other hand, the passive is not related to the pathogen itself. For example, the skin lesions, insect bites, or wounds can disseminate. And this is also another passive way that can basically distribute the pathogens for the tissue invasion. We are going to see that how this active penetration can happen due to production of some of the products by these pathogens. So invasion is a very specific example we are looking at. Once in the circulatory system or the blood vascular system, the bacteria have access to all organs in the system. And the bacteremia is a definition which is a presence of viable bacteria in the blood. On the other hand, the septicemia is another word when you have bacterial or fungal toxin present in the blood, which we commonly known as a septic or sepsis. So this varies among the pathogens. So for example, for Clostridium tetani, which causes a tetanus, these produce a number of virulence factors, but it is non-invasive. So it doesn't go into or destroy the tissues to move from one area to the other. On the other hand, say for example, Bacillus anthracis, which causes the anthrax, and Yersinia pestis, which causes a plague, also produce many virulence factors and are highly invasive. That means they destroy other tissues and gain access to the other tissues. Also, they do not get into the particular cell, but they destroy the tissues for invasion. On the other hand, Streptococcus species span the spectrum of virulence factors and the invasiveness as well. So these are some of the examples of specific invasion of the host tissue. So here in this table, we are looking at some of the more specific virulence factors. What are these? So the first thing that what we are going to see in a pair, the very first one with the coagulase, which is produced by the Staphylococcus aureus, and right here, which is the streptokinase or the staphylokinase. So we are going to connect these two. See that what coagulase does, it coagulates, clots the fibrinogen in the plasma and protects a clot. So essentially in the blood vessels or the side of the blood vessels or the capillaries, these bacteria produce a clot, and inside this blood clots, all these staphylococcus remain. So once they are inside that clot due to the production of coagulase, the immune system of the host cannot access the staphylococcus, and they replicate inside that blood clot, which is basically invisible to the immune system. Now, once they have grown enough inside that coagulated blood clot, they produce an enzyme which is known as, say, for example, staphylokinase for the staphylococcus or streptokinase for the streptococcus. And what they do, that this protein that binds to the plasminogen and activates the production of plasmin and digests the fibrin clots and allows the pathogen to move from the clotted area. So what it does, at the first step, the bacteria produced a clot, divided in the clot. Now it basically dissolves the clot, comes out, and start back again. And this is a way by hide and seek these staphylococcus and the streptococcus inside the blood vessel can move from one area to the other. And this can lead to a particular disease, which is known as a DIC, known as the disseminated intravascular clotting, that can be caused by this coagulase and the kinases produced by either streptococcus or the staphylococcus. Now we are specifically looking at some of them. We talked about the tissue invasion, and some of these bacteria does that. Say collagenase. Then you have these hyaluronidase. So on the other hand, along with that, you have this lecithinase or phospholipase. If you think about that, this collagenase will digest the collagens in the connective tissue space. Lecithinase or phospholipase will digest the plasma membrane of the cells. On the other hand, this hyaluronidase or say elastase and alkaline phosphatase, these will all destroy tissues. So you can see collagenase, elastase, hyaluronidase, lecithinase produced by a variety of different types of bacteria. They can all destroy the connective tissues. On the other hand, there could be a third group where we are looking at these hemolysins, which are produced by a number of bacteria. They will lyse the red blood cells and make it available for the microbial growth. And there is also another one which is known as a leukocidins. These are produced by some of these bacteria, Staphylococcus and the Streptococca, which produce pore-forming exotoxins to kill the white blood cells. So the leukocidins essentially kill the white blood cells, where the hemolysins basically lyse the red blood cells. So you can see that, again, we are not going into the each and every one of them, but gives you an overall idea that what the virulence factors are, that how these virulence factors actually help for the invasion of the host tissues by different organisms. Then we are looking at a very specific type of invasion, which is seen in the Listeria monocytogenes and also Shigella species and Rickettsia. They use this actin-based motility to spread between the host cells. I will post a supplementary video underneath the lecture video, and you can actually see that how these Listeria monocytogenes produce an actin tail behind the bacteria, which propels the bacteria forward, basically. And this not only propels the bacteria inside the cell, it also protrudes this bacteria out of the cell so that that bacteria can be engulfed by the cell which is next to it by the process of phagocytosis. So this protrusion is engulfed by the adjacent cells, allowing the bacteria to enter from one cell to the other. So what is the advantage of that? Once the Listeria gets into the cell, first of all, it escapes the phagocytosis. That detail is not required, but it produces a product known as listeriolysine, which lyses basically the endosomal vesicle, and the bacteria escapes from the endosome before it can get digested by the lysosome. Once they escape into the cytosol, they will produce these actin tails to move around the cell, and then they will protrude out of the cell to get into the next cell. So they are moving from a cell to cell, and they are always inside the cell, but not outside in the tissue space or in the blood. And that's the reason that the immune system gets fooled, that they cannot detect the Listeria monocytogenes easily as they are inside the cell. And the same thing what happens to the Shigella as well as a Rickettsia, because they are always inside the cell, but not outside the cell, where they can be bound to the antibodies and an immune response can go on. So these are some of the particular way that they can move through the tissues. Another example was that we have talked about that the causal organism for syphilis is the Treponema pallidum, which is a spirochete. It bores through the tissues by its corkscrew-like movement. So these are some of the examples of that. And also there are some of the pathogens like the fungi, like Microsporidia. They can produce these polar tubes to get into the tissue. There are some other funguses which also can produce hydrolytic enzymes, which can also destroy the tissues for the invasion as well. So after that, we are looking at that how these different pathogens survive the host defense mechanism. So now let's talk about the overcoming host defenses, or how the pathogens overcome the host defenses. So most of the microbes are eliminated before they can actually cause any disease due to our immune system, and that's how we get rid of most of the pathogens that get inside our body every day. Now, on the other hand, successful pathogens or the primary pathogens overcome the competition and elude the initial host responses as well as the adaptive immune system. So that will give basically the disease. So we are going to talk about more about that, but basically our immune system do have different tiers, where is a tier 1, then you have a tier 2 and a tier 3, and all these needed to be breached and establish an infection. So what are the different types, or what are the different strategies that these pathogens do use to exploit our immune system, or basically fool our immune system? One of them is type 6 secretion system. This is used by many of the bacteria, and you can think about that this is basically a molecular nanomachine. So when these bacteria come in contact with a cell, this secretion system is a complex of proteins which are present on their plasma membrane and the cell wall. It is a transmembrane transcell wall protein system, which is composed of a very complex machinery. And this nanomachine basically inject or transfer the different toxins directly to the cells to cause harm. So this is known as this type 6 secretion system. There are many other type of secretion system, and we're going to talk about those a little bit later as well. Also, they find shelter to avoid the recognition by the defense cells. And you can see that these find the shelter. We talked about this coagulation process, which is of the Staphylococcus or Streptococcus. They also survive and replicate inside the host cell. We talked about the example of Rickettsia, Listeria monocytogenes. They do not come out of the host cells. As they live inside the host cells all the time, they are invisible to the immune systems which are outside the host cells. Also, they squeeze between the host cells. That's also a safe haven where they cannot be reached by the antibodies or the immune system cells. Also, they make capsules to avoid phagocytosis. So essentially, if a bacteria is covered by these capsules, which is called a process which is called a frustrated or aborted phagocytosis. So a phagocytic cell will try to engulf these encapsulated bacteria, and they will abort the process, or they cannot due to the presence of the capsule. And also another example that we talked about, that the Listeria monocytogenes does that once they get into the phagosome or the endosome, they produce something which is called listeriolysin, and they will just breach the membrane of that endosome and come out in the cytosol. So these are some of the processes. They can also burrow under the mucus. Say, for an example, in our gastrointestinal lining, there could be the Helicobacter pylori that can actually get into the mucus layer, into the stomach, and this Helicobacter pylori can survive in the condition. Remember that particularly in the stomach, the pH is probably around 1 or 2 due to the hydrochloric acid secretion. The Helicobacter pylori can live in that pH. The reason being that Helicobacter pylori is not an acidophile. We have talked about those acidophile which can live in moderate to extremely acidic environment. But Helicobacter pylori is a bacteria, which is a pathogen as well, that is not an acidophile, but they burrow under the mucus, and that helps them to avoid the acidity of the stomach, which will otherwise destroy these pathogens. And Helicobacter pylori is one of the causal organism of gastric ulcer, and it can also cause gastric cancer. And they can also secrete this exopolysaccharide to form communal shelter within the biofilms. Classic example for this is a Pseudomonas that can produce these exopolysaccharide, and they cannot be treated by the antibiotics. Even they cannot be accessed by some of these immune cells or the antibodies if they are within these biofilms. Also, they produce different enzymes to inactivate the innate resistance mechanism. We're going to talk about that. Also, they will excrete specialized protein to selectively kill directly the host cells. So we are going to take a look at these. These are directly called the cytotoxins, and we are going to talk about some of these as well. So let's take a look at right here that what are the different things that is being done. So here we are looking at the production of a decoy proteins to bind the neutralizing antibodies. So what we mean by the decoy proteins, pathogens, all of them do have some of the signature proteins on their surface which can be recognized by the neutralizing antibodies. So upon infection, some of these pathogens produce these decoy proteins and secrete it outside. So all these antibodies which were supposed to bind to the pathogen surface, they directly go towards the decoy protein and bind them. That's how they basically are fooled by the pathogen and they can escape. On the other hand, they also can change the cell surface protein by mutation or recombination, or downregulate the level of expression of some of the cell surface proteins. So this is done by the influenza viruses. When a viral infected cell or a bacterial infected cell will produce certain types of molecule on their surface so that the immune system understand that that cell is infected by a pathogen. Now, some of the viruses or bacteria can downregulate the expression of the cell surface protein, and a classic example for that is the influenza virus, as we have said. They can produce some of these capsules that look like the host tissue components, and some bacterial capsules also prevent the deposition of the host complement system and protecting the pathogen from a host defense system. So the host complement system is a wide array. We're going to talk about it somewhere else, but the host complement system is basically a number of proteins which are present in the serum in our blood. These proteins can lyse bacterial cell. Some of the bacterial capsule can prevent the deposition of the host complement system. So this is some of the mechanism by which they can survive. Also, another interesting thing is called a phase variation. So this is basically practiced by the Neisseria gonorrhoeae, which causes the gonorrhea. They alter the pili protein sequence and the expression. So once this Neisseria gonorrhoeae goes into the host, they do have this pilin protein, which makes up the pili. And these are very specific, unique proteins which are recognized by the host defense mechanism or our immune system so that they can surmount a reaction against that, and they produce antibodies or send immune cells to battle these Neisseria gonorrhoeae. But what the Neisseria gonorrhoeae does, that they basically change or alter the sequence of these pilin proteins, and this is known as a phase variation. So they don't look like the pilin protein, and each time in waves or phases, they produce different types of pilin protein. So the defense mechanism or our own immune system completely gets fooled that each time they see that there is something different happening and they don't understand, or the immune system doesn't understand what's going on. And these are some of the evasion processes. Also, there are some other protein that we are going to talk about, produces proteases that degrade the host proteins. These are some of the classic example. On the other hand, say for example, there are some proteins like Staphylococcus produce a protein known as the protein A. So what protein A does, the protein A essentially binds to an antibody, particularly the stalk or Fc region. Don't worry about that. We're going to talk about in a different chapter. But it binds to the antibody stalk region, this protein A of Staphylococcus, and renders it non-functional. So these are some of the processes as well. On the other hand, you can see that particularly to avoid the detection by the host, there are many other processes. Say, for example, special proteins that interferes with the host ability to detect and remove them. Classic example for this one is the protein A produced by the Staphylococcus. On the other hand, for the cause of the host cell fusion, these are examples given by the HIV, then cytomegalovirus, CMV, as well as measles virus. What they do, that once a cell is infected, that cell will fuse with another cell. So basically the virus, without coming out of the cell, they can move from cell to cell by this cell fusion. And this is also another way that they can evade the host immune response. The next one, what we are looking at, that also these pathogens can suppress the host immune response. And how do they do that? They infect the cells of the immune system and diminish their function while ensuring their own survival. Before going into these examples, we can just think about the HIV. The HIV directly attacks the T cells, particularly the CD4 T cells, and these are some of the helper T cells which actually help the immune system to activate. So the activation of the immune system is done by a specific T cell, and HIV directly attacks those cells which are responsible for the immune system to work. And this is how they can suppress the host immune response. The other bacteria like Streptococcus pneumoniae, then Neisseria meningitis, Haemophilus influenzae, these produce a slippery mucoid capsule that prevent the host immune cells to effectively capturing the bacteria. We already talked about this. This is called the abortive or frustrated phagocytosis. Also, eliminate O antigen on lipopolysaccharide to diminish immune response and the clearance. So we are going to essentially take a look at these lipopolysaccharide, and these are particularly present on the gram-negative bacteria where these O antigens are released to diminish the immune responsible. So if you remember that, probably you've heard about that, there is a particular rogue E. coli which caused bloody diarrhea, and it is known as the O157 strain. So what we mean by the O157 strain, that this E. coli is a gram-negative bacteria, so it does have the lipopolysaccharide, and this O157 is the O antigen type 157, and that is what is meant by this O157. Essentially, diminish the immune response and the clearance. So here are some of the examples of the biofilm bacteria. They are protected from the nutrient deprivation, predators, environmental shifts, and antimicrobial agent and the host immune cells. So what are the processes? We'll come to that. Some pathogenic bacteria within the biofilms also exchange the plasmid nutrients. When they exchange plasmid, they can change the virulence factor. Also, they can do a global quorum sensing in the process, and we're going to see that altering the behavior of others. And this biofilm formation make them less sensitive to the antibiotics and more resistant to host defense mechanism. So here is an example. What we're looking at, these are some of these particular bacteria. We call this one as the planktonic bacteria. They are outside the biofilm and just happily moving around. And you can see that they are either directly bound by the antibiotics, or these are the antibodies. They are bound to the antibodies and they will be essentially neutralized. On the other hand, if you can see that right here under the biofilm, you have the same kind of bacteria. And here is a phagocyte which is trying to engulf these. The phagocyte can easily engulf some of these bacteria which are outside the biofilm. When the biofilm is present, there is no phagocytosis of the cells which are essentially inside. There could be some of the dead cells due to the interaction which can happen. These are some of the phagocytic enzymes, but that doesn't cause much harm to most of the community in the biofilm. These are some of the processes by which the biofilm formation negate the effect of antibiotics and the antibacterial antibodies, also the phagocytosis. Next, we are looking at that how the different organisms damage the host. So we are looking at the bacteria first. There are some specific areas which are known as pathogenicity islands, and these pathogenicity islands are large segments, about 100 to 200 kilobases of bacterial chromosome, and also the plasmid DNA found to encode the virulence factor. So what they do, that they increase the bacterial virulence, and these are absent in non-pathogenic members of some genus or species. And they can be spread through the horizontal transfer of virulence genes to the bacteria. Let's get an example of this and how these can work. Say, for an example, there is an E. coli which is known as this O157H7 strain. O157 stands for the O antigen, H stands for a hemagglutinin. This is a pathogenic bacteria which gives bloody diarrhea. It's an E. coli. On the other hand, we have a lot of E. coli in our gastrointestinal tract or the colon, which we can call as a naive E. coli, which doesn't have these bacterial virulence genes in their chromosomes or as a plasmid. So they are beneficial for us, normal microbial flora. They produce vitamins and they help in digestion process, and they are beneficial. We have a mutualistic relationship. Now, somehow a person get infected by this O157H7 strain. This is the virulent strain which contain a plasmid or pathogenicity island either in the chromosome or in the plasmid. So what can happen that once that bacteria gets into the colon, if this pathogenicity island is on a plasmid, this can spread from one bacteria through the horizontal transfer. So basically what is happening that this one rogue bacteria or a few rogue bacteria that the host ingested is now coming in and transferring their virulence factors to the naive bacteria, which were the resident bacteria in the colon. So all your bacteria in the colon, which were all naive bacteria and they were friendly and mutualistic, they now do have this pathogenicity island and they will start producing the toxin, and you will have enormous response or harm, which will cause this bloody diarrhea and cause different types of diseases as well. So these are the benefits of these pathogenicity islands and how the other bacteria can be transformed due to the transfer of this due to the horizontal transmission. So here we are looking at these pathogenicity island. And again, you do not have to memorize any of these names, but you can see this Helicobacter pylori. This is the pathogenicity island, and you have a type 1 pili-secreted proteins which attach hemolysin cytotoxic necrosing factor. So hemolysin will destroy the red blood cells. Cytotoxic necrosing factors will destroy cells, and also uropathogenic protein as well. On the other hand, see that Escherichia coli type 4 secretion proteins, and these type 4 secretion proteins or the type 4 secretion system actually can transfer proteins as well as the DNA or the nucleic acid as well. That's why it's called a type 4 secretion system. There are many different types. They can produce this cytotoxin. We talked about that. And you can see Legionella pneumophilia. This is also another bacteria which can produce this type 4 secretion to the intracellular survival so that they can survive inside a cell. And you have many different things that Salmonella can produce the cytotoxin. Shigella can produce a cytotoxin, and there are many other different ones. Vibrio cholerae can produce these toxin-secreted proteins. And we are going to see a couple of examples and their mechanism of action. So you can easily see that all these different organisms have a specific pathogenicity island which are responsible for producing these gene products and their function. Now, why we call it pathogenicity island, remember that although the outcome is one specific outcome, many times for example, if it is a type 4 secretion proteins, many times a multiple number of proteins are responsible to produce this particular toxin and to secrete that particular toxin. All of these genes are in the pathogenicity island. It is just not one single gene, but a wide array of gene. And that's the reason that we just talked about and why it is so that you have these 10 to 200 kilobases, because of the multiple number of genes that is responsible for producing multiple gene products to cause the pathogenicity. So here we are looking at this toxigenicity. Some microbes possess this toxigenicity or ability to produce these toxins. And these toxins are specific substances that can damage the host. And intoxication is a disease that results from an entry of a specific preformed toxin into the host. So these are some of the definitions. So first, we are looking at the exotoxins, and then we are going to talk about the endotoxins as well. They are both very different. And let's take a look first at what are the different exotoxins. So these exotoxins are soluble in cytosol or soluble in water, and these are heat labile proteins. These are all proteins. That's the most important part. Usually they will get denatured at about 60 to 65 degrees Celsius. They are secreted into the surrounding as the pathogen metabolizes. They often will travel from the site of infection to the other tissue cells where they exert their effects, and usually synthesized by specific bacteria that have toxin genes in their plasmid or prophage DNA, and among the most lethal substances known. And we are going to talk about these, that how lethal these exotoxins can be. So here we are looking at some of the examples of the exotoxins from this chart in your textbook. First, we talked about that these are some of the most potent toxins known to the human beings. So let's take a look at a particular one. If we look at this right here, this is produced by the Clostridium botulinum. This is the botulinum toxin. and it is in a prophage, which is inside the bacteria, and produce an AB toxin, blocks this acetylcholine release, leading to the flaccid paralysis. So this particular botulinum toxin is probably the most toxic known to the human being, which is produced by a natural bacteria. So another also extremely potent toxin is this diphtheria toxin, which is produced by the Corynebacterium diphtheri, which is alters translation, leading to protein synthesis inhibition. So it is known that only just 1 or 2 molecules of this diphtheria toxin is good enough to cause harm. So on the other hand, you can see that right here, the Bacillus anthracis can produce these tripartite AB toxins. And what we mean by these AB toxins, we're going to talk about that. What we mean by that either they are 2 different subunits or 3 different subunits. So tripartite AB toxins do have an EF subunit, which causes the edema or swelling. The LF subunit is a cytotoxin for the cells, and the PA is the B component of the toxin, which is recycled back outside from the cell. So we are going to talk about these. So then you can see Bordetella pertussis produce these AB toxins, Shiga toxins, which is produced by this E. coli O157H7. Remember that we talked about that all the time. This O157 is the O antigen 157 type, and H7 stands for the hemagglutinin H7 type. These are present in the plasmid, so you can easily see that these increases the CAMP level, leading to water secretion from the cell, inhibits protein synthesis, leading to death of the cells. And these can be, as they are in the plasmid, this Shiga toxin can be transferred from these rogue bacteria to your naive E. coli as well. So there are many other examples of that. You can take a look. The cholera toxin is produced by the Vibrio cholerae, and these are all AB toxins as well. And we're going to particularly talk about this in a detail, that how this mechanism works for these. So a lot of these, so Staphylococcus produce this exfoliative toxin, which is a skin peeling, happens due to this. So you can see cytolysin, on the other hand, also leading to water secretion from the cell, increase the cyclic AMP level, and these are all cytotoxin. So different types of toxins produced by a large variety of pathogenic bacteria has been tabulated in this one. So here we are looking at the type of toxins which we just saw in the previous table, that what are these AB toxins. So these AB toxins are produced 2 subunits. A subunit responsible for the toxic effect, and the B subunit binds to the specific target cells. These are specific host site exotoxin. They could be membrane disrupting exotoxins as well. And we'll come to the superantigen in a second. So let's take a look at how these AB toxins work. So here is a cell. This is a host cell. And on right here, there is a specific receptor. And this is the dimeric exotoxin, which has an A subunit and a B subunit. The B subunit is responsible for binding to the specific receptor. This receptor is for something else, which is exploited by this B toxin to bind to that. So this is a clathrin-coated pit, and this will essentially be taken inside. Once upon binding to the receptor, this particular pit is now endocytosed, or receptor-mediated endocytosis will take this inside the cell. And now this endosome will get uncoated. The clathrin is removed. And now all these proton pumps will pump proton into these endosomes, and the pH will drop. At that point, the A and B, with the pH drop, the A and B will separate with the pH drop. The A will now escape from this uncoated vesicle right here, whereas the B component is recycled back onto the outside the cell, and it just goes outside the cell. So this is eliminated out of the cell. On the other hand, the A component goes into the cytosol, and it can do a lot of different things. So here is an example that right here, by the help of this NAD+, which is reduced to N-nicotidamide, and in this process, this EF2, which is the elongation factor 2 of the translation, with the help of this ADP ribosyl EF2 is formed. So once this EF2 forms the ADP ribosyl EF2, this is non-functional, and this will cause inhibition of protein synthesis, and the cell will essentially die. So this is one of the function of these AB toxins, that how it kills the cells or causes the damage to the cell. On the other hand, there could be some specific type of toxins which can insert a preforming exotoxins and insert or make a pore. And if it makes a pore into the plasma membrane of the host cell, you can easily imagine that easily the cytoplasmic content will pass out due to the low osmolarity, whereas the water will enter through this pore, and this can cause swelling of the host cell lysis due to the high osmolarity. Either of these can happen due to the insertion of an exotoxin to form a pore in the membrane. The next will come to the superantigen. So the superantigen is something different where our own immune system get super activated, and that can cause injury or even death. So this particular superantigens are some of the proteins which are produced, or these protein molecules are produced by some of these pathogenic bacteria. They stimulate about 30% of the T cells in the system and cause the T cells to overexpress and release the pro-inflammatory cytokines, and results in a multiple organ failure, and this allows the time for the microbe to disseminate. So how this really works and what this basically means. So here what we are looking at, that this is an antigen presenting cells, and do not have to go into the detail, just an overview, because we are going to talk about this in the immunology chapter about this in much more detail. So what we are looking at here is a macrophage, and we know these macrophages are phagocytic cells. Essentially, they will capture the pathogens and they will present the antigen onto the T cells, and these T helper cells will activate the immune system. And only this happens that if there is an antigen, and here you can see that no antigen being presented. Here there should be an antigen, and only at that time this T helper cell will bind to this antigen and activate the immune system. But what happens, a super antigen binds in between these major histocompatibility complex, which presents an antigen to the T cell. So it links the T cell receptor and the major histocompatibility complex. So when this happens, this T cell thinks that it has been presented by an antigen from here, which is not correct. No antigen has been presented, but it is fooled to believe that an antigen has been presented by the antigen presenting cells. And this fooling is done by this superantigen. So at this time, what will happen, that this particular T helper cells will go out and activate that whole immune system. And this activation of the immune system will cause harm to the body itself, and essentially failure of multiple organs due to this high fever, and this basically can create a shock. This superantigen is responsible for this process. On the other hand, so these are some of the exotoxins that we talked about. And remember that one of the first thing that we talked about, that all the exotoxins are proteins. So they do have a very specific three-dimensional structures. And due to that, many times we do have many vaccines available against these proteins. And those are the reasons that many of these bacterial vaccines are available to us as well, because they are proteins and do have a very specific three dimension. So it's easy to produce a vaccine when you have a protein molecule. On the other hand, now we are going to talk about the endotoxin. This is the lipopolysaccharide in the gram-negative cell wall, can be toxin to specific host. But the whole of the lipopolysaccharide of the outer membrane is not the endotoxin. It's only a part. It is called an endotoxin because it's an endogenous bound to the bacterium and only released when the microorganism lyses or dies. So this is also released during the multiplication. But mainly what can happen, that if your immune system is killing the bacteria, after the bacteria is being killed, these endotoxins can be released. Or also if you're taking an antibiotic that can suddenly killing the bacteria is lysing, it can release the endotoxin and you can have an endotoxic shock. So what is the toxic component? The toxic component is the lipid portion, which is the lipid A. Remember that the lipopolysaccharide has two parts. One is the lipid A, Another is the O polysaccharide. The O polysaccharide do have some antigenic properties that can suppress some of the immune response. On the other hand, this lipid A is the endotoxin. So the lipid A is made up of a lipid, and that's one of the reasons that you cannot make a vaccine against the endotoxin because it is not immunogenic. So these are heat stable. These are toxic to the nanogram amounts, weakly immunogenic. Generally, they are similar despite the sources, means that all the gram-negative bacteria will have a similar kind of lipid A, and they cause the general system effects. What is the general system effects? Once a large number of these bacterial cells are lysed, they can cause fever, shock, damage to the lining of the blood vessels, weakness, diarrhea, inflammation, intestinal hemorrhage, and then fibrinolysis. Fibrinogens can get dissolved, and these are some of the causal organism as well. And ultimately, it can lead to circulation failure. So here we are looking at a septic shock cascade, which can happen that endotoxin can lead to this factor 12, and this factor 12 can do different types of things. It can lead to this coagulation cascade, which is released. It can also trigger this complement cascade. This complement cascade is essentially another serum protein, which is responsible for the immunogenicity. So that will be triggered. Fibrinolytic cascade also will cause different of these fibrinogen type of material, which are responsible for blood clotting. This is a fibrinolytic cascade, and also the kininogen cascade. What are the different things? These will lead to, first of all, this is the most important part. It will lead to release of lots of these cytokines like tumor necrosis factors, lots of this interleukin, platelet activating factor, endorphins, endothelium-derived relaxing factor. And then right here, also, they're going to produce some of these arachidonic acid metabolites. So first, let's take a look at what these things are doing. These will lead to the fever and the shock, first of all, because this is a high alert of the immune system, and that lead to those symptoms. Now, when you have this arachidonic acid metabolism, which is produced these cyclogenosis, leukotrienes, and the prostaglandins, all of these are inflammatory agents. So you will have inflammation everywhere, all throughout the body. Then you have this complement C5a, which will also cause different types of cascade reaction, not going into the detail. Kinine coagulation will cause by this fibrinolytic cascade, and also myocardial depressant substance will reduce essentially the beating of the heart. So you can see that here, this coagulation inside the blood vessels will basically cause the circulatory collapse. So you have myocardial depression and dilation, and the vasculature, vasodilation, constriction, leukocyte aggregation, endothelial cell dysfunction. All of these will undergo into a shock, which is known as a refractory hypotension, which we mean by a refractory hypotension that you have a normal blood volume, but due to this vasculature modification and myocardial depression or dilation, that blood volume is not being circulated, and this leads to the shock. Also, multiple organ failure due to this inflammatory agents that is being working, and that can lead to death as well. Luckily, some of them can recover due to that. So these are some of the things that the overall septic shock cascade. We're not going to ask individuals processes, but just how this endotoxin can be also very dangerous, which is due to the lysing of the gram-negative bacteria and release of the lipid A's. So after that, we are looking at some of the toxins which are produced by the funguses. These are common contaminants of food crops. For example, Aspergillus flavus, parasiticus, produce these aflatoxins, which can cause this chronic and acute liver disease and liver cancer as well. Also, they can contaminate water damaged buildings. Say, for example, this Stachybotrys produce this satratoxin, which are the potent inhibitors for DNA RNA protein synthesis, induce inflammation, disrupt surfactant phospholipid in the lungs, and may lead to pathological changes in the tissues. On the other hand, this is a claviceps purpurea, or the ergot, which is misused by a lot of people, which produce this hallucinogenic lysergic acid, or the LSD, These are some of the mycotoxins. And one of them that we did not talk about, there is also another, which is the death cap mushroom. Now, the death cap mushroom is found in the field, and they actually do have something which is called a phalloidin. This phalloidin is the toxin in the death cap mushroom, which basically stabilizes the actin or the microfilament in our cell, and that also causes acute liver disease, or sometimes we need liver transplant. Almost always death happens from these death cap mushrooms. So this is an idea about the chain of infection, that right from how the transmission, portals of entry, established colonization, establishment, then invasion of the host, then damaging the host, and also escaping from the immune system. So we have an overview of that, how the process of infection goes on by the pathogen, and what are the different strategies that is being used by the pathogen do these harms to a host. So when you work with this material, try to think forward the different types of disease states, using them for examples, and break the chain of infection and control the disease states. This is some of the things that think just as simple as that, just washing hands, or reducing the number of contaminants, or heating or boiling. These are some of the processes by which you can reduce or minimize the infection process. And that will be the end of this chapter.